Ladies and gentlemen, on behalf of InVivoScribe, we welcome you to the webinar organized by the BioIT World Organization. Our program's title today is Complementary Value of Minimal Residual Disease Assessments by Next-Gen Sequencing and Multiparameter Flow Cytometry for the Study of Hematologic Malignancies. In our session today, our speakers will explore various methods, tools, and criteria used to evaluate MRD in hematologic malignancies. This session is being recorded and will be available online. We encourage you to ask questions in real time using the chat feature. We will coordinate getting a response back to you via email. Our first presenter is Dr. Ola Landgren, MD, PhD. He has been the Professor of Medicine and Chief Attending Physician of the Myeloma Service at Memorial Sloan Kettering Cancer Center in New York when the data for this presentation were developed. Dr. Landgren recently took a key position as the inaugural leader of a new research program, Experimental Therapeutics at Sylvester Comprehensive Cancer Center at the University of Miami Health System and the Miller School of Medicine. Dr. Landgren is a world leader in the field of early treatment strategies and molecular and cell-based monitoring of minimal residual disease detection in multiple myeloma and its precursor states. Dr. Landgren is studying molecular mechanisms underlying the trajectory from precursor to full-blown multiple myeloma with the goal to develop treatment strategies aiming to delay, prevent, and ultimately define a cure for multiple myeloma. Our second presenter today is Dr. Swati Shah, MD. She is a diplomate of the American Board of Pathology in Anatomic Clinical Pathology and Hematology. She currently works as the lead hematopathologist of the Flow Cytometry Laboratory at LabPMM located in San Diego, California. LabPMM is part of the InVivoScribe company. Dr. Shah is a board-certified practicing pathologist with 20 years of broad experience in anatomic, clinical pathology, and hematopathology. She has worked at academic and community hospitals as well as high-volume outpatient reference diagnostic laboratory settings in the United States. Her core specialty and passion is hematopathology, with more than 15 years of comprehensive diagnostic hematopathology experience in leukemia and lymphoma diagnosis, as well as MRD testing. Dr. Shah has held various positions as Director of Hematopathology, Assistant Head of Clinical Pathology, and Senior and Lead Hematopathologist. She is a Fellow of the College of American Pathologists and has co-authored a number of peer-reviewed scientific publications. I am now pleased to turn the time over to our first presenter, Dr. Landgren.
Thank you very much for those kind of introductory words, Paul, and thank you very much for inviting me to this seminar. I'd like to thank the audience for attending. So I was asked to share some of my interpretation on the literature and to add also some of our experience as well as looking into the future. So I will dive right into my talk. Before I do that, I will also share with you some of my disclosures. I will give 1 or 2 slides on the background on the role of minimal residual disease in the setting of multiple myeloma, to just put it in a clinical and translational context. Then I will dive into some of the details on our experience working with next-gen sequencing-based MRD testing, and also some of the comparison with flow cytometry that we have. And then the last part will be looking into future directions. So as I'm sure everyone on this call is well aware, people have tried different techniques for years looking into minimal residual disease, both in myeloma as well as other disorders covering cell-based, DNA-based, or imaging-based. And there's a lot of new technology in development as well. Specifically when it comes to multiple myeloma, I think it's important to put this slide on the screen. These are 2 large meta-analyses published in 2016 and 2017, including all the published studies on newly diagnosed patients with multiple myeloma that also contain capture of minimal residual disease detection. And both these meta-analyses show that patients that achieve minimal residual disease negativity have a significantly better progression-free survival and also overall survival. The hazard ratios are in the range of about 0.4 and 0.5 respectively. And that's true for both these analyses. So clearly these are indicators of good outcome. When it comes to advantages and disadvantages of the main methods that are used in clinical practice, as I'm sure everyone on this call also is well aware, and that's also part of these meta-analysis, we have the flow cytometry and we have the next-gen sequencing approaches. For flow cytometry, it's usually available if you have a pathology lab, which most hospitals obviously have in this country, and you can do for send-out. You can do single-cell analysis. It's usually quite quick. And it's quite simple. The problem, though, is that it has to be fresh sample. There are very many ways of doing it, and usually the sensitivity is less than what we require to call it MRD negative, less than 1 cell in 100,000. As for next-gen sequencing, you can bank your sample, and it can be more sensitive, 10 to minus 6 or 1 cell in a million. The disadvantages is that you need to have the diagnostic baseline sample to determine your sequence that you're going to track. And also there is the turnaround time because you have to deal with the extraction of the DNA. And also the fact that because of somatic hypermutation, at least we believe that, there is a failure rate that could go in the range of 5 up to 10%. Of course, also quality of sample will impact that, but that's also true for flow cytometry. Lastly, as a background, there is data from myeloma showing that the more you push down the disease, and if you're able to quantify, the deeper your MRD goes, the longer your progression-free survival is. So there is a significant difference between 1 cell in 100,000 and 1 cell in a million, which is 10 to minus 5 versus 10 to minus 6. So what's our experience working with next-gen sequencing-based assays? We have done all different studies over the years looking at how this relates to myeloma in different aspects. I'm going to share with you a couple of pieces from work that I and my group have worked on. So I start off like very simply here. So we know that the clonotype dominance in skewed light chain repertoire is to be found in patients with myeloma, and that's very different from a normal B cell. Here you have on the left a myeloma cell, and you see how the clonotype dominance is very skewed. And on the right, you have a normal plasma cell from a healthy person. So this, of course, sets the basis for using these assays going forward. What is not 100%, has not been 100% clear in the literature is really to what degree this is true over time. So we know in myeloma that patients can contain a lot of somatic mutations. We know also over time that the disease can change both clinically and genomically over time. So we were doing this study, we published this about a year ago, where we looked over time. So we had 905 patients with myeloma, and we also have healthy control samples. And we looked on the third complementarity determining region, which is part of what we are tracking for VDJ. This is the CD3 region. And then we looked at sequential samples. So one thing we found is that the number of patients with one or more subclones related to the main clone is very, very small. It was 3%. And when we had sequential samples from 45 patients with a total of 101 sequential samples, what we found is that the dominant CD3 sequence remained identical over time. And this is very, very important. This is true even if there is clonal evolution that we can capture with whole genome sequencing. So the bottom line here is that the data strongly support the use of dominant clonal sequences to track over time as reliable biomarkers for long-term tracking of the malignant clone. And this is true even if there is a huge evolution of the disease over time. Another thing that we looked carefully into, and we have known for a long time for very many different assays we have worked on, and this is probably true for any disease, is obviously the quality of the sample. So I want you to pay attention to panel B to begin with. So you have the aspirate smear on the x-axis, and then you have the flow cytometry on the y-axis. So when we talk about plasma cell infiltration, we know that that plays a role for any assay. The more cells you have, the more yield you have. But the point I'm trying to make here is that the number of plasma cells, of course, vary very much based on how we determine the cellularity. So if you look for aspirate smear versus flow cytometry, flow is going to give you a much lower number because you lyse a lot of the cells in the machine. Now, if you look to the left, you compare the aspirate smear on the y-axis to the core biopsy. Again, there is a huge underreporting for the aspirate smear because you have a lot of blood in the sample. In our hands, the core versus the aspirate smear is almost a factor of 2. So 60% core, 30% aspirate smear. And in our hands, the flow could give 1 or 2% because you have so much blood and you lyse the cells. So when we talk about plasma cell percentage, the first question I ask my fellows in clinic, which method? And I always want to see the core because that's where you have the highest number. When we looked at the ability to predict the clonality at the baseline sample in relation to various factors, we spent a lot of time looking not only into plasma cell percentage, we looked at a lot of other factors as well. So I'm going to make this story short. So if you look on the upper left in panel A, you see success versus failure, and on the y-axis you see the plasma cell percentage. So the point there is that more cells, you have a higher success rate. There is no surprise there. Now, if you go down to panel B below, you see that the somatic aberration detected by the capture panel called MyType, there is a much higher clonality detection being successful if there are detectable somatic mutations. So that tells us that if the quality of the sample is better, allowing us to determine somatic mutations, then you're more likely to find the VGJ sequence. There is no surprise there really. But the point I'm making here is that you can have high quality sample with lower plasma cell percentages. And you can unfortunately have higher plasma cell percentages with poor quality. So quality and number, of course, are important factors. And then if you look on the right in panel C, there is a difference in the red line where you have the somatic mutations detectable, yes, so high quality, on left kappa and right lambda. Lambda seems to have a better yield. So those were key findings from this paper, just came out in 2019. So if you look on the very right in the multivariate model, the plasma cell percentage, you have the positivity with the capture panel, and then you have lambda light chain restriction. If you look at the odds ratio, you see that the number of plasma cells is borderline significant, but it's 1.3, while the quality is 6.2 and the lambda light chain is 5.1. So high quality sample is very, very important. And I think in a larger cohort, probably the plasma cell percentage would be significant. But in this sample size, it did not play out. But even in a larger cohort, clearly the quality is more important. So that's something that we need to always remember. Why is lambda light chain restriction more likely to give a yield? Well, so that's something that actually took us back to the basic immunology textbooks where you had to think about that for a little while. And I think this is probably the explanation. So we found mainly clonal Ig kappa rearrangements. Now, the Ig kappa genes in a lambda restricted plasma cell contain a larger number of rearrangements than the kappa restricted counterparts. The lambda light chain expression requires a functional rearrangement of the lambda light chain gene, and that happens after both the Ig kappa alleles have been inactivated. Conversely, the kappa restricted plasma cells by definition have one functional Ig kappa allele. So the second allele could either be germline, which is half the cases about, or it could be inactivated from a deletion or these above mentioned regulatory elements. So you're now talking about 2 to 4 unique Ig kappa rearrangements in the lambda restricted cells, while the kappa restricted typically have 1 or 2. And because the clonality detection is a numbers game, this gives, of course, the advantage to the lambda restricted clones. So I think that is probably why we see that. So that's something that we didn't really think about this, but that's part of our discovery here. What about routine evaluation of MRD using next-gen sequencing compared to high-sensitivity flow cytometry? We spent a lot of time developing in-house assays. When I was in New York at Sloan Kettering, we set up the next-gen sequencing in Maria Arcila's lab, and in Misha Rochal's lab, we set up the flow cytometry. We developed a 10-color panel with a single tube for flow cytometry that has a 50% improved saving of time compared to the old Euroflow method. So we compared the next-gen sequencing to this IGH rearrangement in-house, and we wanted to see for sensitivity and specificity and see to what degree there is correlation. This was done at Sloan Kettering. There were 438 samples from 251 patients. The bottom line with this study is that the index clone is characterized with a success rate of 94%. And similar to what I showed you before, if you have a higher plasma cell cellularity, you can reach as high as 98%. And then if you have lower percentages, that's going to go lower. 85% of the cases were successfully characterized using just a leader and the FR1 primer sets. So framework 1 and the leader, that's 85%. And most of these clones showed high somatic mutation rate. Here you have the capture on the left with the framework 1, the leader, framework 2, 3, IG kappa. And then you see that there is a very small, there's a 2 test failure and no clone detected in 14 cases here. And I think that's probably the hypermutation. And then you see the number of distinct clonal sequences, and you see that the vast majority have 1. So 85% again were leader and framework 1. When we compare the next-gen sequencing to our in-house single-tube assay, and this has been published back in 2017 with Misha Rochal and myself as the first and last author respectively in the Blood Advances, if you're interested in looking this up. And the paper I'm sharing with you now just came out in Journal of Molecular Diagnostics, and it's available online. It shows in the blue bar high concordance for positivity for both methods, and in red, high concordance for negativity. And then there are single-digit numbers where you have positive, negative, negative, positive, and then there is failure. These are very low numbers, 2, 1, 1. So our conclusion is that both these methods work very well. I think one could argue back and forth, but I think a fair assessment is that either of these technologies seem to work very well if you set it up and you know what you're doing and have high volume and you have the algorithm. So it's more a matter of what you feel you want to build on and going forward with. So the concordance is 93%, and the discordant cases, as you saw on the previous slide, are very few. So here you have the last bullet, that I think that the choice of test is more dependent on practical rather than test performance considerations. Maria Arcila has also, as the lead author, and I was one of the co-authors, done this very large study. And I think this is a very powerful publication where she shows the establishment of an IGH change clonality testing in-house, where she characterized B cells and plasma cell neoplasms. On the left in the panel here, you see that this covers the plasma cell neoplasm myelomas, and then you have different subsets of lymphomas from high to low grade lymphomas, and there are also some B-ALLs and hairy cell cases in this as well. This study shows overall that the framework 1 leader, and then you can add framework 2 and 3 as a stepwise approach, has an overall clonality detection of 97%. And 95% in this large series where framework 1 and the leader only. So there is a lot of consistency across the different publications, which of course I think is reassuring, and it kind of reminds me about many years ago when I came here to the United States almost 20 years ago, one of my first mentors was Joe Fromini, and he said, I like to publish the second paper. I always wondered in the beginning, why is that? But I think over time I kind of figure out why he liked that. It's always nice to see that there is consistency and you're not doing these major mistakes. I think this study overall is very important if you are trying to establish an in-house pipeline. You can see a lot about how the procedures and the SOPs are developed. And Maria is a great source if you have questions on how she has set this up. This is also from Maria's paper. It's a little bit more detail. You see that there are 1,189 samples, and it showed 97% clonality detection for all the malignancy types. As part of this study, she also compared to the Biomed2 primers, the consensus primers, and she showed in this paper that using the pipeline has even a better capture than the Biomed2 primers. So again, I turn you to the paper if you're interested, and Maria is the primary source if you have specific questions. I will add some of my future direction perspectives where I do see the field will be going in myeloma. And I'm sure many of you have thought about these things and you have other opinions. That's the beauty of medicine, isn't it? I think we will probably convert a lot of the molecular prognostic work in myeloma towards DNA-based approaches. Here is work that we published also about a year ago, when we did a head-to-head comparison of a next-gen sequencing myeloma-specific capture panel. We compared this to conventional FISH and not karyotyping, but SNP array, which has a higher capture. So FISH and SNP array was the baseline, and that's the denominator from our clinical lab. We had DNA extracted for these patients, and then we ran this panel. So what you're looking at here is you see on the left, the IGH rearrangements, which is found in about 50% usually of myeloma patients, one of these, and they are chromosome 14, usually partnered with 414, 1416, 1114, could also be 1420. And then you have copy number abnormalities, so these are gains and losses, which are frequently found in myeloma. Around 50% of myeloma patients will have one or more of them. So they are so-called hyperdiploid or non-hyperdiploid. And then you have, of course, deletion of 17p, and there are other alterations, 13q as well. The point with this slide is that the next-gen sequencing assay using standard technology as the denominator reveals more than 99% sensitivity and specificity. So I think in this large head-to-head comparison, we have evidence from primary samples that you could migrate over all the testing from FISH and SNP array, or if you use all karyotyping, into DNA-based assay. This assay, because it was myeloma specific, captures the entire IgA's locus, which allows you to replace FISH. If you don't have that, if you only have somatic mutations, or you do a more SNP array-based assay, then you're stuck with FISH because you would not capture the IgA's locus unless you densely position your baits like this assay has. Because it's DNA-based by design, beyond the IgH, beyond the gains and losses, you obviously could look for mutations. So this one has around 120 genes that it covers, and it comes from discovery work through the literature of all the reported mutations you can find in myeloma, and additional also genes in treatment pathways that could presumably lead to mutations as part of resistance mechanism, but that's not 100% known. So why is this important together? I think the importance here is that patient could have, for example, tumor suppressor mutations in the remaining allele in the presence of a deletion on one of the alleles. So this is just for sanity checking. You have 85% of the non-synonymous mutations identified in the samples, total of 362 non-synonymous SNVs. And then you have the K and NRAS around 20 or so percent, BRAF 5%. So this is very similar to the literature. So I just share this with you to justify the data that it seems to be a very reasonable yield. So the samples are reasonable and the assay is reasonable. And you can look this up. It came out in Blood Cancer Journal about a year ago. So coming back to where I think the clinical utility of these assays, what that is. So here you have the tumor suppressor genes. So you have TP53, FAM46C, RB1, and TRAF3, and others. So if you look specifically, for example, at patients with a 17p deletion, in this series, there were 29 patients. Now, in those 29 patients, 13 of them, which is 45%, they actually carried a mutation of the non-deleted allele. Now we have the biallelic inactivation of TP53. One is deleted, the other one is mutant. And there are actually, as we would expect, I think there are 3 cases that are mutant only in the absence of a deletion, but that's only one of the alleles. So 13 cases were biallelic inactivation. And this is work from Brian Walker published in 2019 on the left PFS, on the right OS. The biallelic inactivation has a significantly worse progression-free and overall survival. The monoallelic, which again refers to either deleted or mutant only, has the same clinical outcome for PFS and OS as you have for a wild type. So a patient without a 17p deletion has the same PFS and OS as a patient who has only a deletion 17 in the absence of mutation. And in the standard clinical setting, if we read a FISH, we would not know what the other allele is. So we probably, I think it's fair to say, we probably tell 50% of the patients you have high-risk disease, and they don't. The ones who really have the bad outcome, the biallelic, we don't really know who they are and how they are different from the other patients at the clinical setting. The last part of this wrap-up here is we then took the data from this same assay, and we ran the data through a algorithm called the mixer algorithm. And this is a algorithm that allowed us to reconstruct the VDJ region from this short read data. We had 74 patients. These are primary samples from this series. And we had also worked them up with the LymphoTrack assay. We have done the leader and framework 1, and then as a second step, framework 2 and framework 3. When we compare the LymphoTrack results with the mixer VDJ short read data, we found the same sequence in 70 out of the 75 samples. So that's 95%. So I think this sets the stage that you could think of going forward a next-gen sequencing-based profile of myeloma to replace FISH and SNP array. And you could also then capture somatic mutations. And you could also have capture of VDJ clonotypes. I'm not saying that this is ready for clinical use right now, but I'm looking into the future here. I'm trying to be a little bit provocative. I think this really sets the stage for long-term tracking. So I think it's possible to have a molecular lab. So whether this should be done in a single assay or it should be done in a bundle way, you could do capture of mutations, you could do capture of IGH, you could do capture of gains and losses, and you could do MRD separately. Maybe the MRD assay has to be separate because you would not find every single case. Here, for full transparency, we already knew the sequence for VDJ when we used the mixer. So what would we find if we just blindly look into this with the mixer algorithm? That's something I don't know. So I don't want to oversell that approach. But I think, to me, there is data to support that we are heading into a molecular approach. And if we are thinking of using a single platform for baseline, I still think that the same sequence, even if that's true, could still be used for MRD tracking for longitudinal samples. And of course, there could be a lot of other approaches as well, and I'm sure Many of you probably are thinking, why don't we do a single cell or circulating DNA in peripheral blood or other approaches? And of course, we are interested in these things also. The future, I think, is going to be very exciting and a lot of change for sure. So with that, I would like to thank you so much for your attention. And I think the questions, if you may have any questions, should be communicated by email. I'm happy to answer those. Thank you so much.
Good morning, everybody. Thank you for this opportunity for presentation at this webinar. My talk will emphasize the following points. MRD assessment for acute myeloid leukemia using multi-parameter flow cytometry for MRD assessment, AML MRD recommendations from ELN as well as NCCN, and screening and MRD AML panels at Lab PMM in ViperScribe. So why MRD is important? Minimal residual disease tests are routinely used in hematologic malignancies, especially routinely in CML and pediatric ALL, and are also increasingly evaluated in multiple myeloma, AML, CLL, and other lymphoproliferative disorders. And several methods of MRD assessment are available in clinical and research settings. When you look at MRD testing, the testing method chosen is important, and there are key factors that go into consideration of the testing method chosen, especially the assay sensitivity, assay specificity, reproducibility of the assay, despite the timing, location, operator, as well as the standardization of this assay, which could be verified with methods such as FDA clearance or CLIA certification, and the test impact on the patient, especially based on the sample type, amount of the sample needed, ease of the sample procurement, quality of the sample, availability of the test, and also financial impact on the patient as to the cost and insurance verification. There are various methods of MRD testing available and have been used in clinical and research settings. They are morphology, immunophenotypic analysis, karyotyping, fluorescent in situ hybridization or FISH, polymerase chain reaction, PCR, and next generation sequencing, NGS. Morphology is based on detection of leukemic cells with the aid of microscope on bone marrow or peripheral blood samples, which has limited sensitivity but has been used in clinical practice for many years. Immunophenotypic analysis is based on using immunochemistry or flow cytometry to identify markers or antigens on the cell surfaces, nucleus, or cytoplasm of malignant cells. Karyotyping is based on chromosomal analysis, which has been used in diagnostic workup as well as risk stratification, but has low sensitivity and also can be used in up to 50% of the patients. FISH also has high specificity, but also again is applicable in less than 50% of the patients. PCR-based methods are highly sensitive, but again, could be applied in limited number of patients. Next generation sequencing can reach a very high sensitivity up to 10⁻⁻⁶ levels, but is expensive and needs complex bioinformatic pipeline and complex interpretation, and may not be available at all the settings. MRD assessment has been used in myeloid malignancies, especially in CML and AML, especially acute promyelocytic leukemia. And the testing is used to identify the presence or absence of residual disease using specific molecular and genetic markers, and has been increasingly looked at in AMLs other than those without PML-RARA translocation. So I will discuss the MRD testing with focus on acute myeloid leukemia now. Acute myeloid leukemia, or AML, is a clonal neoplastic proliferation of hematopoietic precursor cells causing excessive accumulation of myeloblast and other immature myeloid precursors. The diagnostic work is based on morphologic assessment, including identification and quantification of blast percentage, immunophenotyping by flow cytometry for lineage determination, and also the remission is confirmed by morphology when less than 5% blast are seen in the bone marrow sample. The prognosis of AML is dependent on many factors, but significant weight has been placed on genetic and cytogenetic factors. Despite improvement in characterization and prognostication and treatment, approximately 50% of the AML patients will relapse after initial therapy. Traditionally, MRD assessments have been performed using morphologic and imaging techniques, which have limited sensitivity. And despite having achieved a complete remission, a patient may harbor up to 10⁻⁻10 persistent leukemic cells at level undetectable by conventional morphologic assessment. Hence, highly sensitive methods such as multi-parameter flow cytometry and molecular testings are required for risk stratification for early recognition of impending relapse and direct subsequent therapy. The accompanying image shows the relationship between the amount of persistent disease in patient and the rate and the duration of relapse. The lower the disease burden, the later is the relapse and less chances of relapse. Hence, MRD testing with sensitive assay is important. The NCCN guideline recent version indicates that the patient who achieve complete remission by morphologic assessment alone can harbor a large number of leukemic cells in the bone marrow. MRD in AML refers to presence of leukemic cells below the threshold of detection by conventional morphologic methods. MRD is a component of patient evaluation over the course of sequential therapy, and role of MRD in prognosis and treatment is evolving, and participation in clinical trials is encouraged. What is the significance of MRD in AML? MRD detection is an emerging prognostic marker. Numerous studies have shown the power of MRD in AML in predicting risk of relapse and overall survival. In the setting of allogeneic stem cell transplant, those who proceed to transplant with MRD positivity have been shown to have a risk of relapse and 3-year survival similar to those with active AML disease. Hence, prognostic criteria have now expanded to suggest inclusion of MRD as an outcome measure. The most current methods for MRD assessment in AML include molecular techniques, including quantitative PCR and NGS, and multiparameter flow cytometric analysis, often with two related approaches, leukemia-associated immunophenotype, LAIP or LAP, and different from normal DFN approach. And this publication by Dix and colleagues also show that in patients with morphologic remission, patients who have MRD positivity, the 3-year relapse rate is about 70% and 3-year overall survival of 25%. And those with MRD negative status, the 3-year relapse rate is 30% and 3-year overall survival of 35%. And more sensitive methods are being developed, some of which are described here, which I can go into details at a later time. Flow cytometry methods for MRD detection are based on detection or combination of cell surface markers present in leukemic blasts but not on normal hematopoietic stem cells. The multi-parameter flow cytometry has advantage over PCR methods that it can be applicable to over 90% of AML patients. Two approaches are used in multi-parameter flow cytometric analysis of MRD, as I described, leukemia-associated immunophenotypes, LAP, or different from normal DFN approach. Those who are not familiar with flow cytometry, this is a schematic representation of a flow cytometer in which cell suspension is injected into flow cytometer with single cell file of cells, goes through the fluidics. They encounter a laser light which gets refracted and collected by different dichroic mirrors, and if the cells are conjugated with different types of fluorochromes, they emit light which are passed through various filters, and the signal is produced which is analyzed by a computer. So what is lab approach? In lab approach of MRD testing by flow cytometry, it involves identification of aberrant expression of markers or marker combination on leukemic cells, which are identified at the diagnostic samples and are followed in subsequent follow-up samples. Labs are based on identification of aberrant expression, which could be cross-lineage antigen expression by myeloid cells, such as lymphoid expression, lymphoid antigen expression such as CD7, CD19 on myeloid blast, asynchronous marker expressions on blast, under or over expression of certain markers on the blast, which helps them to be identified amongst the normal hematopoietic cells in the sample. This approach is widely used for tumor immunophenotyping at diagnosis and can be followed in subsequent samples. The advantages is rapid turnaround time, which results can be available within 24 hours, but it requires a fresh sample and cannot be performed on archival samples. And conventional flow cytometry is less sensitive than multicolor next generation flow assays. When you have flow assay using more than 8 colors, it can detect one cancer cell in 10⁴ to 10⁵ cells assessed. This approach is difficult to standardize in multiple multi-center settings, and interpretation requires great technical expertise. The different from normal approach uses standardized combination of antibodies, and that is applied to all MRD analysis, which allows identification of markers outside the spectrum of maturation process of normal stem cells. This approach can be applied without the knowledge of leukemic-associated immunophenotypes that are present in diagnostic samples, and is able to detect any immunophenotypic shift that occurs during the course of the disease or treatment in a patient. Again, inherent to flow cytometry, the assays are difficult to standardize in multi-center settings. And compared to lab-based approach, DFN may have lower sensitivity. A trial using both flow cytometry methods, lab and DFN approaches, found a lower percentage of MRD positive patients in clinical remission using DFN at 24% compared to those found using lab at 34 to 42%. The limitations of flow cytometric methods for MRD detection AML is lack of standardization assay between the laboratories, subjective interpretation involved in multiparametric flow cytometric analysis. The poor quality of sample and hemodilution can interfere with the specificity of MRD analysis and reduce its sensitivity. And sometimes the flow cytometric assay may have MRD negative status, but patients can still relapse, which could be due to presence of leukemic stem cells, LSCs in a patient, which are undetectable by routine methods. Hence, the ELN recommends inclusion of a tube in the assay to determine residual leukemic stem cells in multi-flow cytometric MRD analysis. This table shows the comparison of multi-parameter flow cytometry and NGS-based MRD assays. Both by lab and DFN approaches have overall lower sensitivity compared to NGS, which can reach up to 10⁶ or 1 in million cell sensitivity for detection, as opposed to 10⁵ to 10⁵ by a multi-parameter flow cytometric analysis. The advantage of flow cytometry is it can be applied to significant number of patients, up to 90% compared to NGS. The lab needs diagnostic sample in order to follow in the subsequent analysis. DFN can be used without information about the diagnostic sample and can detect the immunophenotypic shifts or emerging abnormalities. The disadvantage of flow, it needs significant experience, knowledge of immunophenotypic patterns of normal marrow in order to detect the abnormalities present in patient samples. The NGS disadvantages is it's expensive, time-consuming, and technology may not be widely available and has complex need for complex interpretation. The ELN consensus guideline published in 2018 consensus guideline on MRD assessment in AML, and they state that MRD monitoring should be considered as a part of standard care for AML patients. And ELN guidelines recommend harmonization of both lab and DFN approaches for flow cytometric assessment for MRD. And they recommend first pool bone marrow sample with minimal hemodilution and sample to be processed as soon as possible after collection. And ELN guidelines recommend using a cutoff of 0.1% between MRD positive and MRD negative for interpretation. And they recommend between using 500,000 to 1 million viable cells to be included in the analysis. And interpretation should be done by operators with extensive experience in MRD testing. The ELN guidelines on MRD testing intervals indicate that the MRD should be assessed with multiparameter flow cytometry for subtypes other than APL, CBF, AML, and NPM mutated AML, and the patients with mutant NPM, NPM1, RUNX1, RUNX1T1, CBFB, MyH11, and PML RARA. ML should have molecular assessment of the residual disease at the informative clinical time point, and they have guidelines for MRD testing during the treatment phase and follow-up phase as described in this table. And they also have guidelines, clinical guidelines for MRD assessment, and they indicate that the failure to achieve MRD negative complete remission or rising MRD levels during or after therapy are associated with disease relapse and inferior outcome and should prompt consideration of change in the therapy. MRD should be assessed with pre-transplant and post-transplant time points. All clinical trials should require molecular and or multiparameter flow cytometry assessment of MRD at all times of evaluation of response, and monitoring beyond 2 years of follow-up should be individually decided based on the relapse risk. Clinical outcome based on MRD status using multiparameter flow cytometry has been evaluated in various studies, and few are shown here. The Hoan-Sack AML42A study was predictive, has shown that multiparameter flow cytometry MRD assessment was predictive and independent of other prognostic markers, and in particular in ELN intermediate risk group where the treatment decision can be difficult. And this study showed that MRD positivity after cycle 2 was associated with a high risk of relapse within 4 years. Relapse-free survival was 23% and relapse incidence of 72% compared to 52 and 42% for MRD negative patients. This next study by Konke and colleagues also looked at MRD status by flow cytometry in very early treatment, which would enable the treatment intensification as soon as possible. And they assessed the prognostic value of multiparameter flow cytometry MRD during aplasia on day 16 to 18 of induction therapy, and they found that the MRD positivity at this stage can be an independent predictive of poor outcome with 5-year relapse-free survival of 16% compared to 43% in MRD negative patients. And this is a recent review by Dix et al. Talks about measurable residual disease in acute myeloid leukemia using flow cytometry, the review of where we are and where we are going, and is an excellent paper to have as a guide for current status. And this again table shows clinical outcomes using multiparameter flow cytometry separately as well as combined with NGS MRD, and using both lab and DFN approach of multiparameter flow cytometry assessment for MRD, and they show significant correlation with relapse rate, either with both NGS and flow cytometry together or independently. When both are positive, it shows a high relapse rate of 73%. When both are negative, 4-year relapse rate of 26%. And with the DFN and NGS combined assessment, again, when both are positive, the 18-month relapse incidence was 37% and overall survival of 48%, versus both negative, the relapse rate was significantly lower. So they either together or independent are very useful in MRD assessment. And in order to increase the sensitivity of multiparameter flow cytometry assay, various strategies have been recommended as to including assessment for leukemic stem cells, including machine learning algorithms and mass cytometry time of flight. And lastly, I would like to bring to attention the flow cytometric capability at LabPMM and in VivoScribe. In early 2021, LabPMM will offer complementary MRD assessment with multiparameter flow cytometry and NGS. Currently, we have a fully functional flow cytometry lab at San Diego location with a CAP-CLIA standard validated hematolymphoid screening panel that can screen and diagnose majority of the acute myeloid leukemia and lymphoid malignancies. And it's a 10-color 4-tube panel, and as described here, and shows the representative sample of our analysis template and some gating strategies here. And this is a representative report that is created after hematopathologist review. We are in the process of optimization AML MRD panel by flow cytometry based on recommendation by the ELN MRD working group consensus recommendation. It will be a 4-tube panel covering myeloid monocytic markers, some leukemic stem cell markers, and some key markers for detection of aberrancies. We plan to acquire 500,000 CD45 positive events using sample metrics of bone marrow in EDTA and sodium heparin. And we'll be using the lab-based DFN approach for tracking the MRD. Again, I described these in detail in my previous discussion, lab and DFN approaches. And the reporting for AML MRD by flow cytometry would follow the recommendations by the ELN working group on MRD. We will be using a 0.1% cutoff as a threshold for diagnosing MRD positive versus MRD negative, and various parameters that can be described in the report will be viability, total CD45 positive events for all the tubes, the blast percentage, the gating strategy used for detection of blast, the lab as a percentage of blast and WBCs, and if there is a multiple labs detected, all frequencies will be added up, and description of immunophenotypes, and interpretation based on the level of MRD detected. If it's above 0.1% will be called positive, between less than 0.1% to more than 0.01%, it will be a MRD detectable and quantifiable but of uncertain significance. And when in cases when there is complete absence of aberrancies, it will be called MRD negative. And in certain cases, if interpretation was not possible, it will be called multiparameter flow cytometry not possible. And again, there will be comment about the sample integrity, viability, and list of markers analyzed. In conclusion, MRD assessment in AML, the importance is that AML is a heterogeneous malignancy that requires active MRD surveillance due to ascertain deep remission. Multiparameter flow cytometry and NGS-based MRD assessment offers synergistic benefits. Multiparameter flow cytometry MRD assessment can be done with two approaches, lab and DFN. And next generation sequencing MRD assessment can ensure accurate MRD assessment over time, and ultra deep sequencing can achieve sensitivities of about 1 in 1 million. And MRD is rapidly becoming a standard in AML care because it's the most powerful predictor of relapse. And these are the key references that one can look at on this topic. Thank you for this opportunity, and thank you for your attention.